Well, thank you to Steve for the lovely introduction, and thank you to everyone for coming. Can the people in the back hear me? Okay, great. So today I'm going to tell you about the work that I've done in our larval zebrafish model that we use to investigate lipid metabolism. So just to start, you may have noticed one term in my very long title that is important. It's dietary fatty acids. So I just want to give you some really basic background on what I mean when I'm talking about dietary fatty acids. So fatty acids are part of the lipid category of molecules. Here's one right here. Each bend in this line in this molecular diagram is a carbon, and carbons form 4 bonds, so where there's another line between carbons, that's a carbon-carbon bond, and when you don't see lines drawn, all of those spaces are filled up with hydrogens. And in this particular fatty acid, this is a saturated fatty acid, palmitate. It's 16 carbons, and all of the available bonds that aren't forming a single carbon-carbon bond are filled up with hydrogens, and that's why we call it a saturated fatty acid, because its capacity for carrying hydrogen is saturated. So there are multiple categories of fatty acids that we have to consider when we talk about the contents of food, and this triglyceride molecule here, I'm showing you 3 different categories. So here's a saturated fatty acid again. This is a monounsaturated fatty acid, and that means that it has one carbon-carbon bond that has 2 bonds between the carbons. So there are 2 hydrogens that are basically missing from this molecule in places where they could be. And if a fatty acid has multiple double bonds between carbons, so even less hydrogen involved, that's a polyunsaturated fatty acid. And those of you who might not be very familiar with this field have probably still heard of these fatty acid categories because unsaturated fatty acids are associated with positive outcomes for cardiovascular health. So there's a lot of evidence that variations in fatty acid content of the diet does correlate with differences in health outcomes. Now, dietary fatty acids don't really arrive in that single fatty acid molecule form that I showed you. In food, dietary fatty acids are actually incorporated into complex lipids, such as triglycerides, cholesterol esters, and phospholipids. So here I've put boxes around the fatty acid components of each of these molecules. Additionally, cholesterol itself, which does not carry a fatty acid, is also part of the general lipid mixture in a typical diet consumed by humans. So when we eat any of these complex lipids, the fatty acids are released from these molecules in the intestine when they're broken down by digestive enzymes. So just as an example, I'm showing you the breakdown of a triglyceride. So here, this triglyceride will get broken down into 2 fatty acids and 1 monoglyceride, which is the middle fatty acid molecule here, plus this glycerol group at the top, at the left side that you see. These smaller molecules can then be taken up by enterocytes, which are the cells in the intestine that absorb and process nutrients. And I'll be talking about what goes on in these cells a lot in the rest of this talk. Now, once fatty acids arrive in the enterocytes, they have to immediately be reincorporated into complex lipids, such as triglycerides or the other categories that I showed you. Free fatty acids are not good for the health of cells. They can get into membranes and mess up membrane integrity and function. So healthy cell will resynthesize complex lipids from any fatty acids it takes up. These complex lipids can then either be stored for a little while within the enterocyte in a lipid droplet structure, and I'll show you what those look like later, or they can be packaged into lipoproteins, which are lipid and protein structures that carry lipids into the circulation and throughout the body so that they can get dispersed where they need to go. So when I'm talking about where dietary fatty acids go, there are a couple of different senses of where that I'm going to be discussing. There are spatial destinations, so what organs, what cells, what organelles do they end up in? There are also biochemical destinations. So does a fatty acid get oxidized for energy and released as the waste product CO2 that we exhale, or is it stored later into complex lipids? And within this category, where do these complex lipids go in the cell, and what complex lipids are made? So in general, fatty acids can end up in cellular structures. They can be used to build cell membranes, which are made of phospholipid, or they can be stored within cells. Different tissues can be used to store lipids. Adipose tissue is what we basically consider fat tissue. This is a healthy place for lipid storage to go, but other tissues can store lipids as well. For example, the liver, which I'll show you some pictures of this later, can store large amounts of lipid, as can muscle tissue, including the heart. However, this is not associated with positive health outcomes, and there's a phenomenon called lipotoxicity that is associated with overload of lipid within these tissues. So these are all different concepts that need to be considered when we're talking about the fates of dietary fatty acids. Now, what I'm going to concentrate on are the biochemical destinations of dietary fatty acids. And this is a process that is determined just as they're absorbed into the intestine. So this is an image taken by Juliana Cardin, a previous grad student in the Farber lab, of the intestinal enterocytes of a live larval zebrafish. And this fish has been fed a fluorescent fatty acid that's being incorporated into lipid in these cells. And here you can see an outline of an enterocyte and these round structures, which are lipid droplets. So the contents of these lipid droplets consist of triglycerides, cholesterol esters, and phospholipids that would form the membrane of the lipid droplet. And the reason that we care about whether a dietary fatty acid ends up in any of these three categories of molecules is because the destination of a fatty acid determines its capacity to influence cellular physiology. Another way to say this is that some lipids are more bioactive, more physiologically active than others. So triglycerides and cholesterol esters are basically just storage centers for fatty acids until they're held in these structures until they can be burned for energy or until they can be used to synthesize other lipids later. Phospholipids, however, build the structures of the cell. They build the plasma membrane, they build the organelle membranes, so they're out there in physical contact with the rest of the components of the cell, and they're interacting with membrane proteins, so they have a lot more potential to influence cellular function. Additionally, fatty acids are also used to make bioactive molecules such as signaling lipids. They can modify proteins, and they can be involved in cellular damage, such as the effect of having too much free fatty acid that I alluded to earlier. So just to go a little bit more into a basic example of how cellular fatty acid composition can influence physiology, this is probably the most basic example that we go over in biology courses and textbooks, is that the amount of unsaturated fatty acid in the cell membrane influences its fluidity, which means how freely can the proteins that are sitting in that cellular membrane move around. So a cell membrane containing more unsaturated fatty acid tells on the phospholipids will be more fluid than one that's more saturated. So when we go to study effects of individual dietary fatty acids, traditionally this has been done using observational studies. You've probably all read about observational studies of human diets that contain more polyunsaturated fatty acids being better for cardiovascular health, and more saturated fatty acids generally being less healthy. These studies can also be conducted by feeding, say, defined diets to mice and looking at health and disease outcomes. So once researchers would discover a particular health outcome that was correlated with an increase in a particular type of fatty acid in the diet, you would want to follow this up by examining the metabolism of a specific fatty acid. And traditionally, a lot of this research has been done by delivering just a single fatty acid to a cell culture. However, that is not really a realistic model of how lipid metabolism works for a lot of situations. Food is a mixed nutrient and a mixed lipid substance. Cells in a living animal are never just metabolizing one nutrient at the same time, and they're never just even metabolizing one type of fatty acid at the same time. Here I have some fatty acid compositions of a lot of these 6 vegetable oils on the bottom and some other foods that are basically just straight fat, or the 3 on the top. And just in case you can't see the text, the green is monounsaturated fatty acids, the orange is saturated, and the blue is polyunsaturated. And you can see that even in foods that are just entirely fat, the composition of the different fatty acids is very different between them. And when within each of these categories, there are also hundreds of different possibilities for the individual molecules. So the way to feed an animal or a model system a realistic mixed lipid diet, but to still look at the metabolism of an individual nutrient, is accomplished through metabolic labeling. So here I'm showing you a representation of a mixed lipid meal, and then this molecule in the middle is containing a label. This could be most commonly fluorescent or radioactive, and basically you would feed an animal this mixture, and then you would look at the products and the destinations of this particular molecule and what's being synthesized from it with a detection system that can pick up this tag. So radioactivity has been widely used, and it's probably the most physiologically similar way to accomplish metabolic labeling. However, you cannot see radioactive molecules under a microscope. So what our lab does is we actually use fluorescent fatty acids as metabolic labels. So there's a lot of precedent for using fluorescent fatty acids in live imaging in a variety of models. So this is the fluorescent fatty acid called BDIPC12. It's labeling lipid droplets in C. elegans and Schistosoma mansoni, which are two different worm model systems. And this is done by live imaging. And we've also been using this for a while in the Farber lab to deliver these fluorescent fatty acids to larval zebrafish and use live imaging to see where they end up in the body. And these fluorescent fatty acids are applicable to cell culture as well. This is again the same fatty acid, BDIPC12 labeling lipid droplets in a cultured humans, in an isolated human cell. So this is the basic setup for delivering fluorescent fatty acids as metabolic labels to the larval zebrafish. And we raise the larval zebrafish to 6 days old, and at this point, this larva, it's about 5 millimeters long, and you can see that it's optically clear, so it's suited for live imaging. But at this point, it's ready to start eating. So it's got a fully functional digestive system. It has a liver, intestine, pancreas, gut microbiota. So it's ready to consume its first meal at this point. We mix one of these fluorescent fatty acids, or a couple other types I'll show you later, into a meal of 5% chicken egg yolk. This is a mixed lipid diet, and larval zebrafish really like to eat it, so it's a good delivery method. And then we can allow them to metabolize this for a period of 0 to 24 hours after feeding, at which point we have 2 readouts from the same experiment. So using one metabolic label, we can do live imaging and we can do biochemistry, which is great for getting a big picture understanding of the metabolic processes that are affecting dietary fatty acids. So some work done by Juliana Cardin shows that these 3 fluorescent fatty acids, C16, C12, and C5, and these numbers are referring to the length of the saturated carbon chain, she was able to show that these are incorporated into complex lipids when they're delivered with a mixed lipid diet. So when you feed them to fish without egg yolk, you really just get the substrate back. So these bands on this TLC plate represent these free fatty acids that were fed to the fish. However, you start seeing a lot of other bands when you feed them in a mixed lipid diet. So here we see cholesterol, ester, and triglyceride products from all 3 of these fluorescent fatty acids, and she was also able to observe phospholipids from these 2. However, TLC is not really an ideal readout for a metabolic labeling experiment, because in a lot of systems, you want to be able to detect changes in individual lipid species. And each of these bands up here in the triglyceride range, this is all of the triglycerides. Each of these bands represents, I'd say, from my research, I know there's about 30 different fluorescent triglycerides in each of these bands, and of the possibilities that could be synthesized, there are actually tens of thousands. And the same is true of the phospholipid bands. So this system allows you to detect changes in entire classes of lipids, but doesn't allow you to detect changes in specific lipids. So our goal for developing a better metabolic labeling system, we wanted something that could resolve and quantitate individual fluorescent complex lipids, and we wanted to be able to also understand how different fluorescent labels affect metabolism. Because I've just been showing you this one green fluorescent label, but that's not the whole picture. There are a lot of different fluorescent lipids out there, so I wanted to get metabolic profiles of basically everything that people use in live imaging just to see how we could better compare results of live imaging from different experiments. And finally, I wanted to apply this system to understand how nutritional context affects fluorescent fatty acid metabolism. So how is the metabolism of these fluorescent fatty acids we use as metabolic labels affected by the food that it's delivered to larval zebrafish in? So in order to begin doing this, we developed a high-performance liquid chromatography system. So HPLC is a technique that separates mixtures for quantitative analysis, and our system is adapted specialized for use with lipids. So basically, what we do is that we have a column that it's a nonpolar substance that lipids that are extracted from a tissue sample will bind to. We run a series of solvents over this column, and over 2 hours, the solvent mixture gradually changes from a more polar substance to a more nonpolar substance. So this means that the components of the mixed lipid extract are all going to load onto the column at the same time, but they're all going to get washed off at different times, including according to their chemical properties, specifically by solubility in the solvent mixture. So we have two detectors. We have a fluorescent detector that will pick up the fluorescent lipids that are being synthesized from the fluorescent fatty acids we give to the larval zebrafish. And then we have a charged aerosol detector, which is going to pick up all of the lipids. And this is, I want to point out, this isn't a lipid detector. This is more, it's basically an everything detector. So it's going to pick up everything that's coming off of the column from our samples. Now I'll be showing you a lot of these chromatograms. This is the output from the HPLC machine. And the peak area, which corresponds roughly to peak height, is the quantity of the molecule or set of molecules represented by each of these peaks. And the retention time, which is its position on the x-axis of this diagram, corresponds to its identity. So in order to identify one of these peaks, we have to purchase a standard that matches it and show that it runs at the same retention time, or we can use our mass spec experiment that Steve told you a little bit about earlier. So I'll get to the metabolic labeling experiments in a few minutes, but I wanted to show you a few things that we've been able to learn from our analysis of the total lipid content of a larval zebrafish. So in this experiment, I fed two groups of larval zebrafish either a high-fat, high-cholesterol meal, which is chicken egg yolk, or I fed them a standard meal, which is the fish food that we give our larval fish in the nursery in the fish facility downstairs. And this is a low-fat, low-cholesterol meal. And I give them time to digest this food and for their intestinal contents to clear out, and then I extract the total body lipids. And what you can see is that after a single high-fat, high-cholesterol meal, the cholesterol content is the same. The cholesterol ester content is the same. That's this peak and then this doublet right here. But the triglycerides are increased tenfold. And this is just from one meal in the larval zebrafish. So this was really encouraging because it meant that we could do these short-term, high-throughput experiments in our larval zebrafish model. So the way that I identified the contents of each of these peaks by mass spec is that we took our existing machine, we reproduced our chromatography, so we ran the same experiment that produces this lipid profile, but ran it into a mass spec machine instead of our charged aerosol detector. And we used this single experiment to inform us about the contents of each of these peaks that we observe on our lipid profiles. So by doing one mass spec experiment without actually having to buy the machine, we can then get a lot more data out of the HPLC experiments that we're able to run at this facility. So to get from the mass spec data to the contents of each of these peaks, I basically lined up the retention times on this CAD trace with the retention times I got from the mass spec. I binned these peaks into different categories by retention time, and I was able to see that each of these triglyceride peaks, for instance, consists mainly of 2 to 3 different triglyceride species that run at the same point. So there are actually, I'd say 10 to 100 components of each of these peaks, but most of them are minor. 90% of the area of each of these peaks is accounted for by 2 to 3 triglyceride species. So this was pretty interesting, and this is what we use to interpret our data that we get from this equipment now without having to run mass spec every time. So moving on to the metabolic labeling, when we run the fluorescent detector and the charged aerosol detector, we're able to get total lipid profiles and fluorescent lipid profiles out of the same sample, which means that we can get a lot of data from one experiment. So to orient you to these chromatograms, this area of the chromatogram is phospholipid, and that's on both of these traces, except for this tall peak, which is cholesterol. And then from this point over are the nonpolar lipids, so triglycerides and cholesterol esters. And one thing that I want to point out is that our fluorescence detector is very, very sensitive. So I've marked a peak on each of these traces that represents a point that's kind of in the middle of the linear range for detection and quantitation. And you can see that we've got about 100,000-fold sensitivity in the fluorescence detector over the charged aerosol detector. And this is important in case there are concerns about the metabolic label that we add to our food mixture changing the overall metabolic physiology itself. It means that we can use a very, very tiny amount of metabolic label and still get a product profile out of it. So wanted to show you the first bit of data that we got from these metabolic labeling experiments. Here I'm showing that the chain length of a fluorescent saturated fatty acid does affect its partitioning into complex lipids. So This is our short chain labeled fatty acid called BDPC5, and this ring structure on the end is a green fluorescent tag, and then we have a 5-carbon saturated fatty acid. And it makes a few phospholipids, which are in this blue portion of the chromatogram, and then a wide range of triglycerides and a single cholesterol ester. This is also basically the description of what happens with our longer chain fatty acid, BDPC16. BDPC12, shown in the middle here, forms a more diverse profile where it actually makes a good deal of phospholipid. So our hypothesis as to why this occurs is that BDPC12 probably most closely resembles the fatty acids that are most common in food that a larval zebrafish or any other animal would encounter out of these three. This molecule is a lot shorter than what we would expect, and this molecule, BDPC16, is a lot longer. You'll notice this fluorescent tag is quite large. Now, the most common fatty acid in animals is palmitate, which is 16 carbons. And what we think is happening is that this 12-carbon fatty acid plus this fluorescent tag, which is about 4 carbons long, is being seen by the enzymes in the cells as something that's pretty close to palmitate. Now, we wanted to test this out. So what I did was I moved the HPLC machine into the radiation room, and I ran the lipid extracts from fish that I had fed a mixture of BDP-fluor labeled C12 and radioactive palmitate through the HPLC to analyze them. And I was able to get fluorescent and radioactive product profiles out of the same fish. So these two were being metabolized in equal molar amounts at the same time. And they make a similar range of products. So the radioactive palmitate products are in orange, and you'll see that there's a large hill here that's in the phospholipid area. And here's some quantitative data. And at the same time, BDP-fluor C12 also makes a pretty large amount of polar lipid, which just isn't true of the other two fluorescent fatty acids that we have available to us. So moving forward in our experiments, we considered BDP-fluor C12 to be the best available option. Even though it's not identical to radioactive palmitate metabolism, it forms a significant amount of phospholipid, so it's the closest, and this is what we consider our standard lipid for metabolic labeling experiments at this point. So, in order to investigate the effect of different colors of fluorescent tags on the metabolism of fluorescent fatty acids, this is an experiment that Meredith Wilson and I did together, and here you can see lipid droplets in the larval zebrafish liver. So in this experiment, Meredith took green and red fluorescent labeled C12 fatty acid and fed it to larval zebrafish in an egg yolk at the same time, and did live confocal imaging of the liver. And you can see in the overlaid image, the yellow shows the overlap between the two colors. There's significant co-localization. So in terms of spatial deposition, the products of these two fluorescent fatty acids end up in the same places. However, when we take biochemical analysis from the same experiment, we can show that they make a similar range of products, but the proportions of the products are different. In extracts from the same number of larvae, it appears that the green fluorescent fatty acid is metabolized into products a lot more readily than the red fluorescent fatty acid, and we also do get more phospholipid and more cholesterol ester relative to the rest of the products. And this makes sense that it would be a little bit more difficult for cells to handle the red fluorescent fatty acid because the fluorescent tag is even larger on the red version than it is on the green version. You can see we have this big ring structure where a methyl group is just on the green. So it is metabolized, but it just doesn't happen as readily, and the product profile is different. Something that's much more important to note is that when you deliver these fluorescent fatty acids in a standard diet, so this is a lot lower lipid than chicken egg yolk, the green BDP-C12 is still metabolized into a pretty diverse range of products. We have phospholipids and a few triglycerides and the cholesterol ester over here. And the red phospholipid doesn't really make much, fatty acid, doesn't really make much phospholipid anymore, and we only get a few nonpolar products. So if somebody is looking to do imaging using red fluorescence and they want to feed a standard diet, this would be something they would need to know about. And this is just an example of how our metabolic labeling experiments can influence what you do in live imaging. Yes?
So in order to see these fluorescence, is
Mm-hmm.
what if whatever that has to be the lipid that doesn't have the diamond, but it's still there, it would be detected?
So you actually do detect the BDP dye. When you do these live imaging experiments, we can get a back.
Oh.
Oh. Yes.
But what if the dye would cut off?
Mhm.
The remainder that's becoming incorporated coming from the didn't see it.
So I guess for the dye group to be cut off by itself, just because if the fatty acid is getting oxidized, then it's going to get oxidized from the opposite end. So we do expect to see some free BDIPY dye. It doesn't end up in the trace. Unfortunately, in these HPLC traces, it would be all running off in the solvent front. So if there is free BDIPY, it's there because the fatty acid attached to it has gotten oxidized completely and it's just gone. As far as the label just falling off by some other mechanism, yeah, then we would essentially have C12 that was just floating around the cell and not acting as a label anymore. I guess, yeah. So that is a concern, and that might be something that could be something that happens more readily with the red fluorescent lipid, and it could be one reason it's not metabolized as well anymore. It wouldn't. I don't think that would necessarily interfere with the type of results just beyond just making the signal weaker. So in conclusion, this is just a short summary of what I was doing in this method development project. But I just wanted to summarize to say that I've learned that carbon chain length influences metabolism of fluorescent fatty acids. Total dietary lipid content influences their metabolism. And in general, the green BDIPY tag is probably better for live imaging than the red BDIPY tag, although red BDIPY lipids are still going to be useful because they still do form products. So the question that I wanted to answer using these methods I started to develop was getting back to the general question of how nutritional context affects the metabolism of fatty acids. So I decided to investigate how dietary cholesterol availability affects fluorescent fatty acid metabolism. So this is a basic summary of what we know about how dietary cholesterol and dietary fatty acids interact. And one way they interact is by being combined together into cholesterol esters. So cholesterol is absorbed into enterocytes through the NPC1L1 transporter. Fatty acids have their own transporters, or some of them can actually just diffuse through the enterocyte membrane. Both of these molecules go to the endoplasmic reticulum, where they encounter the enzyme ACAT2, which combines them to make a cholesterol ester, which is a nonpolar neutral lipid storage molecule. And these can end up in lipid droplets, or they can be exported throughout the rest of the body in lipoproteins. So one major question in the field is how enterocytes regulate esterification of cholesterol. And the predominant hypothesis right now is that this enzyme ACAT2 that sits in the ER membrane is sensing the amount of cholesterol in the ER membrane. And because membrane function is very sensitive to the amount of cholesterol, cholesterol is a regulator of membrane fluidity and rigidity. If there's too much cholesterol in the ER membrane, it's going to be necessary to get rid of it to keep the cell functional. So ACAT will link up cholesterol with this acyl-CoA, which is an activated dietary fatty acid. It means it's ready to form a bond to another molecule. It'll put it into a cholesterol ester, and it'll package it out into a storage structure. So I wanted to see what would happen if I blocked dietary cholesterol uptake. So if there's no cholesterol coming in from the intestine, how does that influence what the cell does with dietary fatty acids? And to do this, I used the drug ezetimibe. This is actually used in humans to treat high cholesterol. It's known as Zetia. So I used a very similar experiment to the ones that I've shown you before to investigate how blocking dietary cholesterol uptake affects the metabolism of dietary fatty acids. I treated larval zebrafish with ezetimibe both before, during, and after feeding them a BDIPY fatty acid in a high-fat, high-cholesterol diet. And then I used HPLC to compare the products of this fluorescent fatty acid, and this is BDIPY C12. And I noticed that, the only lipid species that undergoes a change between these two groups is that a lot less cholesterol ester of BDIPY C12 is formed when the fish are treated with ezetimibe. So that's this peak labeled with the arrow right here. You can see it's a lot shorter relative to its neighbors in the bottom trace. Furthermore, I performed some experiments where I did the same feeding and drug treatment, and I dissected the intestines out of the larval zebrafish and extracted the lipids from these, and I was able to show that ezetimibe acts in the intestine. So this cholesterol ester peak disappears in the intestinal samples in the ezetimibe treated group. So something that's notable, I went through and I did this experiment with all of the fluorescent fatty acids that I've shown you products of so far, the 3 different lengths of green fluorescent fatty acid plus the red BDIPC12. And I got a significant reduction in the amount of cholesterol ester synthesized when I treated them with ezetimibe, the cholesterol absorption inhibitor. And this result is actually similar in magnitude to what you get when you treat larval zebrafish with a ACAT inhibitor, which interferes directly with the action of the enzyme that makes cholesterol ester. So first of all, this may seem to not be a very big deal. You reduce the availability of cholesterol, you get less of one of its products. However, cholesterol is very abundant in larval zebrafish. So cholesterol is the largest, tallest peak in the traces from larvae that have been fed egg yolk or larvae that have been fed a low cholesterol meal. And in fact, not only does a high-fat, high-cholesterol meal not affect the total amount of cholesterol in the fish, it's also not affected by giving them ezetimibe or by giving them the ACAT inhibitor. So there's a lot of cholesterol around. So it's actually quite interesting that cholesterol esterification seems to be primarily regulated by the availability of incoming dietary cholesterol when it happens in the enterocytes. So there is some precedent for thinking that dietary cholesterol and dietary fatty acid interact. These are some results from 1969 showing that feeding triglyceride with cholesterol promotes the uptake of cholesterol in the intestines of rats. And these are some results obtained by Oscar Reyes in the Farber lab, showing that dietary fatty acids are necessary for dietary cholesterol uptake in the larval zebrafish. So this is the intestinal lumen of a larval zebrafish that's been fed top fluor, which is another fluorescent tag labeled cholesterol in egg white, which doesn't contain any fatty acids. And all of the fluorescence is just sitting in the intestine. It's not getting absorbed by the enterocytes. But when he feeds fluorescent cholesterol with a small amount of oleic acid, which is a monounsaturated fatty acid, then we see cholesterol signal getting into the enterocytes, so it's being taken up. So I think that these results, combined with my own, suggest that the dietary cholesterol absorption and esterification may be coupled, so that there's something about the transport processes when cholesterol and fatty acids enter the cell and they interact at the ER at the same time, and this would be the primary driver of how much cholesterol ester gets made from dietary fatty acids. So moving on, I wanted to apply the techniques that I've designed to studying the problem of alcoholic fatty liver disease. So just to give you some background on AFLD. This is a progressive condition, and these are some histology slides of what a liver looks like at various stages of this disease. So here's a healthy liver, and this is a fatty liver, also known as steatosis. This is a very common condition, and it's largely asymptomatic. It's 15 to 30% of the U.S. population, according to the NIH, and 70 to 90% of the obese population has excessive amounts of triglyceride in their livers. So this doesn't really cause problems on its own, but it can progress to steatohepatitis, which is the combination of a fatty liver with inflammation. And if this condition goes on for long enough, then it can progress to cirrhosis. Cirrhosis is a more serious condition that can begin to impact liver function. It's inflammation, fatty liver, and fibrosis. So scar tissue starts to form, and it negatively affects how the liver works. And cirrhosis is also a precursor condition for liver cancers. So I do want to say one thing that we don't understand about this disease progression is that only some people with each of these conditions will go on to develop the next condition in the progression, and it's not perfectly correlated with, say, the amount or duration of alcohol abuse. So I wanted to use my model system to investigate how diet might play a role. So zebrafish are an established model for alcoholic fatty liver disease. These are some images of the livers of larval zebrafish that were exposed to a 1% ethanol solution over a long period of time. At 2 weeks, they start developing steatosis, and by 12 weeks, they started developing some inflammation and fibrosis. So these two images on the right are stained for collagen, which is the main component of the scar tissue that forms, and you can see a lot more of it in the ethanol-treated fish. The larval zebrafish is also a model for acute alcoholic liver injury. So these are some images from the Sadler lab who have done a lot of work with the larval zebrafish and ethanol. And you can see that when they're treated with ethanol overnight, there's a lot more lipid in the liver. So this is oil red O. It's a stain that labels neutral lipid, and here the liver is bright red in the treated larva, and you can barely even see it in the untreated ones. So what I wanted to investigate was whether or not dietary fatty acids contribute to steatosis that results from this acute ethanol treatment in the larval zebrafish. So I took larvae that were 1 day older so I could feed them. I treated them with ethanol overnight, and then I fed them. This is imaging with BDP-C5 in chicken egg yolk, and I imaged the larval zebrafish after they'd had a few hours to metabolize them. And you can see there's a lot more labeled lipid droplets and labeled structures in the ethanol-treated larvae than in the untreated group. And just to show you some quantitation, we're getting in terms of total fluorescence, we're getting pretty big fold change in the amount of dietary lipid products that end up in the liver. And I've been able to show that this effect persists for about 20 hours. So this actually is relevant to humans, despite what I told you about alcoholic liver disease being a progressive condition. Humans also do exhibit acute alcoholic steatosis, or acute alcoholic liver injury. So this is from an experiment that was done in 1968 in the Lieber lab, and Charles Lieber was one of the pioneers of alcohol metabolism research. And he basically had some human volunteers who were either light drinkers or non-drinkers, and he gave them, I think it was 2 drinks an hour for 12 hours. And he actually said in the paper that this was his model of typical weekend binge drinking, which might tell you something about the 1960s in alcohol culture. But then he took liver biopsies. And after two days of this binge drinking model, you'd see a lot more fat in their livers, and he also saw inflammation occurring acutely in about half of the subjects. So these people didn't drink for 2 weeks afterwards. They got more biopsies taken, and this effect cleared away. But this does show you that there are some metabolic changes going on even after just acute alcoholic exposure. We do see an interaction with lipid metabolism. So some questions in the field about the interactions of diet and alcoholic fatty liver disease is whether or not the composition of a fatty liver in terms of the fatty acid composition of what's being stored in those hepatocyte lipid droplets, how might this contribute to the variability in fatty liver disease outcomes? And the question I chose to investigate was whether or not ethanol influences how dietary lipids are metabolized beyond just causing more of them to be deposited in the liver. So I'll show you what I've found so far in order to address this question. So we're going back to this basic experimental paradigm where we can feed these fluorescent fatty acids as metabolic labels to the larval zebrafish and examine them with HPLC. And a striking result that I want to show you is that ethanol does alter the metabolism of the fluorescent fatty acid BDP-4C12, which is we consider our palmitate analog. There are more changes that you can see through quantitation, but one thing I observed right away from looking at the chromatogram is that in the ethanol-treated larvae, we get an increase in this lipid species, which I've identified by mass spec as PC, so this is a common membrane phospholipid that's carrying BDP-4C12 and oleic acid, which is a monounsaturated fatty acid that's common in membranes. The one that runs right next to it, which is BDP-C12 plus palmitic acid on a phosphatidylcholine, doesn't change. And when I quantitate the entire lipid classes of total phospholipid and total triglyceride over a time course of metabolism of BDP-C12, I'm able to see that we get a significant increase in phospholipids in the ethanol-treated larvae at 4 of these earlier time points. So this is going up to 16 hours after feeding, and we also see a similar decrease in triglyceride at some of the same time points. So I also see this when I feed BDP-C5, our short chain fatty acid. So in the ethanol pretreated larvae that were given 1% ethanol before feeding, we get a big increase in this peak without seeing any changes in any of the peaks in the rest of the HPLC trace. And this, again, is a phosphatidylcholine that contains an unsaturated fatty acid and our fluorescent lipid. So when I quantitate this, again, I see a 1.9-fold increase in this phospholipid of interest, and I don't see an increase in the phosphatidylcholine containing palmitate and our fluorescent fatty acid, and I also don't see changes in the 2 other phospholipids that are formed from BDP-C5. So why is this interesting? What could this possibly mean for cellular physiology? So in summary, you'll notice that ethanol is increasing the incorporation of dietary fatty acids into phospholipids, specifically into phospholipids that have that dietary fatty acid plus an unsaturated fatty acid. And we're also seeing a decrease in total triglyceride. So there is a precedent for ethanol increasing the unsaturated fatty acid content of membranes in cell culture experiments, and also for it increasing membrane fluidity. However, the way that it might interact with dietary fatty acids to do this has been unexamined so far. So this is something that I'm really excited about following up. Now, one thing that I want to point out, you may have noticed we get acute hepatic steatosis and less triglyceride synthesis from dietary fatty acids in the same model. This may seem contradictory, but what I want to point out to you is that the larval zebrafish liver is tiny compared to the rest of the body. It is, when we look at a whole body lipid composition on an HPLC trace, the liver accounts for a negligible portion of it. So what I think is going on here is that we're seeing more triglyceride or more cholesterol ester in the liver and less in other tissues. So they're making less of it, but it's being pulled into the liver. It's also possible that the fluorescence in these hepatic lipid droplets is largely accounted for by phospholipid. And if I go back and do some imaging with these type of experiment with better resolution, I might be able to start to try to answer that question. It's also possible, I only did this experiments with C5, and it's possible that the decrease in triglyceride doesn't occur with this metabolic label as it does with C12. So I am going to repeat these experiments because I need to redo the images in a way that will allow me to investigate effects on lipid droplet size and number. And the resolution I got with these didn't allow me to do automatic image analysis. So hopefully I'll be able to investigate these questions further. Now, as far as how ethanol metabolism might alter lipid metabolism, just looking into mechanisms. These are the two metabolic pathways by which animals process and detoxify ethanol. So there's the one that kicks in first is alcohol dehydrogenase, which metabolizes ethanol to acetaldehyde, which is a toxic byproduct, so this has to be converted to acetate. Acetate is actually a precursor for fatty acid synthesis, so this is one of the ways that ethanol could contribute to steatosis is by being made into fatty acids itself. The second pathway, when the alcohol dehydrogenase enzyme is overloaded, is this is microsomal ethanol oxidizing system. This is also called the CYP2E1 pathway. And this is a molecule that actually metabolizes a lot of different things, not just ethanol. But it does take over metabolizing ethanol when ADH is overloaded, and this pathway generates a lot of oxidative stress. It also converts ethanol to acetaldehyde that has to be converted to acetate. So ways that this might damage cells, oxidative stress can cause widespread membrane damage, which could result in higher membrane turnover, so the cells would have to make more phospholipid to replace it. Acetaldehyde can also form adducts with various components of the cells, so it might be disabling the function of enzymes that are necessary for cellular function. One thing I want to point out about the larval zebrafish model is that they actually skip this step. So I think this is the reason that we see acute effects of ethanol in terms of metabolism in the larval zebrafish at a stage where they're still able to swim around and eat. So that makes them a great model for looking at the combination of ethanol metabolism and metabolism of food, because they essentially don't show any neurological or behavioral effects at an ethanol dose that causes them to have a metabolic phenotype. So that's something that's really good about this model. So we're basically just skipping to this step. So in order to start asking the question of how ethanol might drive dietary fatty acids into phospholipid, there are a couple of mechanisms I could consider. It could be upregulating remodeling of existing phospholipids somehow. So there's this enzyme PLA2 that can cut one of the fatty acid tails off of a phospholipid, which then could be replaced by a dietary fatty acid. This is activated by inflammation and oxidative stress, which we know is occurring with ethanol exposure, so that's a possibility. And I could look at an enzyme activity assay for this. I also want to consider, are these phospholipids being made de novo, or are they being made from scratch? And because I'm seeing changes in phosphatidylcholine, a way I could do this is to give larval zebrafish radioactive choline and see if they incorporate more of it if they've been treated with ethanol beforehand. So that's an experiment I intend to do soon. Another question I want to ask is, where are these phospholipids? Are they overturning at the cell membrane, at organelles, or both? And I want to do subcellular fractionation on larval zebrafish samples to answer this question. So I wanted to remind you as to why changes in phospholipid are interesting is that phospholipids are bioactive lipids. And when they're incorporated into membranes of the cell membrane or organelles, they can affect membrane stability and fluidity, protein function, for example, the ability of immune system receptors such as TLR4 to dimerize and transmit their signals, and they can also be involved in signaling themselves. So the hypothesis that I've gotten from this is that ethanol amplifies the effects of dietary fatty acids by pushing them into more bioactive lipid species. And this is interesting because it means that variations in diet composition could contribute to variations in alcoholic fatty liver disease outcomes, either by putting lipids into phospholipids that promote cellular health or that exacerbate the effects of ethanol. Also outside of the context of alcoholic fatty liver disease, understanding interactions between ethanol metabolism and lipid metabolism could be important for non-alcoholic fatty liver disease. Because non-alcoholic fatty liver disease, which is common in obesity and metabolic syndrome, does not necessarily exclude people who drink moderate or small amounts of ethanol, so understanding how that component of their diet is influencing their disease process is also important. And finally, I want to leave you with, this is very far off, but I think that my results might apply to possibly explaining diet-dependent potential benefits of alcohol. So you've already all heard about the Mediterranean diet effect and how it does incorporate small amounts of ethanol consumption. And if ethanol is taking healthy fats in the diet and pushing them into places in the cell, phospholipids, where they can have more of a physiological effect, then maybe that explains the role of wine consumption along with healthy fats in the promotion of cardiovascular health by the Mediterranean diet. So, want to know, does anybody have any questions? Yes. Oh, yes. So we don't know yet. It's an ongoing experiment. It's possible. The possibilities we're considering is that the fatty acids physically interact with NPC-101, the cholesterol transporter. But yeah, it's a topic of ongoing work in the lab, and that's really I'm showing you what we've got right now. Yes.
Have you looked at lipid profiles elsewhere in the fish to see if there's like distant effects?
So I intend to do experiments with adults. Now that I know that we do have a adult chronic alcoholic liver disease model, I was going to dissect tissues. Because I was pretty disappointed that I can't really get a lipid profile from the larval liver. It's just too small, so experiments with adults will help answer that question. And I could look at adipose tissue too. Larvae, at the stages I'm working with, they don't have adipose tissue, which is obviously important in lipid metabolism, so investigating that in adults will be interesting. All right, well, I wanted to move on to my acknowledgments. And I do have the traditional slideshow, but I still have this up so far. So first, I do want to thank my thesis committee, especially David Cohen for traveling all the way out here to see my talk. They've been very helpful in keeping me on track, and Steve alluded to us doing some kind of crazy experiments, and I feel like the rest of the committee was instrumental in keeping things from getting too crazy and making sure I actually eventually graduate. So I really want to thank all of you for that. I do want to thank the fish facility staff. I'm a little bit different in terms of zebrafish research in that you may have noticed I'm not using any transgenic animals. I don't spend a lot of time in the fish facility, which means that I'm working with other people's fish or fish that are shared by the lab. So I'm very grateful to all of the people who have helped maintain them to make my experiments possible. Wanted to thank the people at Thermo Fisher Scientific, Joseph Rizica, for basically teaching me mass spec for a week, even though I don't think that's actually part of his job, and Mark Plant for helping us get started on our HPLC method development. I want to thank, of course, the lab. Everyone has been wonderfully supportive. I want to thank Steve for giving me a chance to do method development, which is I know risky, but it's something that I really like doing, and it's given me a lot of clarity for what I want to do as a career. So, don't really have any, I realized this morning, I don't really have any good photos of the lab together, so I've had to settle for this one of Carnegie at the Science March, and most of them are in there somewhere. So I do want to thank all of you for participating in that with me. I want to thank my classmates for being so supportive. That's not all of them, but again, I don't actually have any good pictures of all of you either. I see some of you here. Hi, thank you for coming. I want to thank the BioWise crew, so Val, Steve, Rob, and Chandra, not pictured, at the Science March. I've had some wonderful experiences volunteering with them and getting to get out in the community and educate people about model organisms and animal development, and it's been a wonderful opportunity to be able to answer questions about what we do, and also to just get an idea of what people know and what people don't know when it comes to things like nutrition or how scientists operate. I also do want to thank all of my theater friends, one of whom you might recognize as Becca, who is also a Carnegie friend. I have slowly been trying to rope her into doing student theater with me, and she's an excellent actress. And I do want to thank the biology department for inspiring me as a playwright. You may not know this, I've started writing plays, but I actually got my start writing the retreat skits every year. So this is my classmates and I doing our Real Scientists of Carnegie retreat skit, where we did a reality show parody at the Carnegie Institution, and I'm so grateful that the department has a culture where we can do things like this. It's great. It's great for morale, and yeah, I'm very happy that we do all this stuff. I also want to thank my friends from back home and my family. Specifically my parents, who are right here. They've always encouraged me to get into sciences from a young age, getting me a microscope and a chemistry set and all of the books that I wanted to read about everything. And yeah, I just want to thank them for their support. They've been incredible. And of course, no Carnegie thesis talk would be complete without pet pictures, so here's Sugar and Amiga. I would also like to thank them for keeping me sane. So, and that's extra slides if anybody asks questions. So yeah, thank you for coming.